So, Dr. Bhattacharya, thank you so much for welcoming us here to the Waisman Center today. It's been a fascinating day of learning the various projects that are underway, and it's been a great opportunity to get to meet you in person. I know that you were one of the first to really begin to develop induced pluripotent stem cell lines as that technique became available. Could you just talk for a moment about the role of induced pluripotent stem cells as a model for investigating Down syndrome and other disabilities?
Sure, and I'm so happy that you came to the Waisman Center today. The induced pluripotent stem cells, I think, really open up a whole new field for both Down syndrome research, but also research on really any disorder or disease, because they allow us to make disorder-specific stem cells, or with a very specific genetic mutation, and take those stem cells and turn them into, theoretically, any cell type in the body. And I think for Down syndrome, that's particularly helpful because the trisomy 21 affects almost every organ system to some extent. And so while we work on using induced pluripotent stem cells to turn them into brain cells and learn about how the brain develops and how those neurons function, they can also be used to study heart development or blood development. And I think that they're just really invaluable for Down syndrome research. The other thing that I think is so great about them is it gives you human cells to work on. The extra chromosome 21 is a complicated genetic mutation that can't be completely recapitulated in an animal model because of the structure of the chromosomes. And so by having these induced pluripotent stem cells, we have human cells that have just the extra chromosome 21 triplicated, or just the addition of the extra chromosome 21, and that allows us to study the human chromosomes in a human cell. And so I think that is the other really valuable aspect of induced pluripotent stem cells.
Good. Some people who see this may not understand the difference between embryonic stem cells and induced pluripotent stem cells. Can you just clarify that point?
Sure. So human embryonic stem cells are derived from human embryo about a week after fertilization. And because of that, they have a lot of ethical controversy surrounding them. And so in the time since human embryonic stem cells were first isolated, scientists spent a lot of time trying to figure out what is so special about a stem cell. What is it that makes a stem cell a stem cell? And more importantly, can we turn other cells into stem cells that don't have so much controversy? And so that's where induced pluripotent stem cells came from. Scientists learned that you can take a fully differentiated cell, like a skin cell, and basically turn back the clock and make it have the characteristics of a human embryonic stem cell, although it comes from a skin cell. So it's not, it shouldn't be ethically controversial, and you can get skin cells from many different people with many different genetic mutations.
So you can come to understand or explore anyway the variety that exists within the Down syndrome community, for example.
Correct. So you can, as we talked about, study differences that are due to the extra chromosome and not differences that are due to different people.
Now, as we begin to hear more and more about clinical trials beginning on drug therapies and more drugs in the pipeline, potentially, what about the value of these cell lines for investigating the potential of molecules?
So I think that they're very valuable for that because, as many of us know, most drugs that are developed for clinical trials and for the market are tested on animals and then go straight into people. And human induced pluripotent stem cells and the cells that we make from them provide a bridge between animal models and people. And so at a minimum, it allows us to test whether certain drugs may be toxic to human cells, even though they might not have been toxic to mouse or rat cells. And so I think that's a whole new field I think that will be opening, is using these human-induced pluripotent stem cells for drug testing and toxicity testing.
And I suppose it's a really speculative question, but we've heard in the media often that, especially around adult stem cells or embryonic stem cells, that they're being introduced into humans to regrow certain tissues. Do do you see the potential for using induced pluripotent stem cells for therapeutic treatments at some point, or are they mainly a good model for researchers to use to investigate neurodegeneration or the use of pharmacological products?
I think it depends on what you're studying. I think something like a neurodegenerative disease where a specific population of cells is lost, such as in amyotrophic lateral sclerosis or ALS or Parkinson's disease, I think there the idea of cell replacement is really attractive. But I think in other conditions where we really don't still know what is it that is different, or what steps go awry during development, to know what to replace. And so I personally don't think that cell replacement is a good option, at least at this point, for many of the neurodevelopmental disorders such as Down syndrome.
Good. Well, thank you so much for your time today. And again, it's been a great pleasure to meet you in person and to see what all is going on here. Best of luck in the future, and we look very forward to staying in touch.
Great. Thank you. Thank you.
Thanks again.
I really enjoyed your visit.